Hi dear students, welcome to the session on vectors. Today we're going to talk about vector. What is a vector? As we had mentioned in the previous classes of recombinant DNA technology, vector is a vehicle which carries your what? Your gene of interest. Now what is the speciality of this vector? Or what is the speciality of this vehicle which will carry your gene of interest? If it has to carry a required gene of interest, it has to be capable of removing some element of its DNA, giving place to a foreign DNA. So, if something has to be a vector, or if it is has to be capable of carrying a foreign DNA, it should follow certain features. Now, what are they? Usually, we choose vectors which are of small size because if they are being found to be of low molecular weight, it becomes easy to handle, and moreover, the chance of shearing of the vector DNA is being found to be very less. Now, you should remember even the vector which we consider here, or the vehicle which we use to transport a small segment of DNA into the host cell, that vehicle which we consider here is also a DNA. And that DNA should be of low molecular weight. It should be present in multiple copies, and it should have less chance of having more than one restriction site of the same restriction enzyme. That is, if you consider echo R1 is a restriction enzyme, and you have a vector consider okay, fine, I'll just put a C. Consider this is a vector or a vector here, and this vector should be in such a way that it should contain only one restriction enzyme's cut site for the same restriction enzyme. That is, if I consider the echo R1, okay, the echo R1 site should be present only at one particular site, okay. It shouldn't be present at many sites of the particular vector. Now, if I go to consider a BAM H1 site or something like that, it should be present on the vector only at one particular point, but not at two different points of the vector. Now, if you consider this case, you have the restriction if consider these two are echo R1s. If you have two echo R1s sites on the particular vector, it is not a good feature. So if it has to be a vector, it should not have more than one restriction site of the same restriction enzyme. I hope you got it. That is, if you have echo R1, it should have only one echo R1 site. That would be more better rather than having two echo R1 sites on the same vector. And you can see that we also require that a vector should always code for a selectable marker. Now, what do you mean by a selectable marker? The most best example of a vector is a plasmid DNA. And some plasmids are what? Coding for certain antibiotics. So, if a cell has that particular plasmid, the cell also gets the property of antibiotic resistance encoded by that particular plasmid. In such a case, the antibiotic resistance is being considered to be as a selectable marker for that particular plasmid. So, if a vector, as I told you, a plasmid is a good example for a vector. So, if any molecule of a DNA or could be served as a vector, then it should have a selectable marker on it. It could be different things. It could be antibiotic resistance, or it could be the presence of an enzyme which will help in the detection of it, or anything like that. So it should have a selectable marker on it, or it should encode or it should code for a property which is being found to be selectable. And of course, a vector need to have multiple restriction enzyme sites for various restriction enzymes. Okay, so various restriction enzymes are needed because only if it is able to be cut by various restriction enzymes that we can cut that portion of the vector and incorporate into a foreign DNA into it. The next feature that we say is the presence of an origin of replication. Now, what is an origin of replication? Origin of replication is something which will help to replicate the DNA. Only if the vector has its own origin of replication, multiple copies of that particular vector can be created. So, the presence of the origin of replication is also an essential feature of a vector. So, I hope you got an understanding what is a vector. A vector is a vehicle which is being used in a recombinant DNA technology or genetic engineering to carry a foreign DNA. And usually, to be a vector, it should be of low molecular weight, easy to handle, and less chance of shearing would be there. And when it is being considered to be low molecular weight, it has less chance of having more than one restriction enzyme site for the same restriction enzyme. And always, to be a vector, the vector should code for a selectable marker, and it should have multiple restriction sites for various restriction enzymes, and also an origin of replication which will enable its replication. If you go to consider the vectors, you can divide them into various types. You can divide them firstly as cloning vectors and expression vectors. What are cloning vectors? When the purpose of your genetic engineering is to just clone or to produce a clone of a particular gene or to know a particular sequence and all that. In such cases, cloning vectors are used. But if you do expect to express a particular protein at a very high level in the host cell, then you use a expression vector. I'm not going to the details right now. We'll be going in the next classes regarding what are the details of cloning as well as the expression vectors. And on the basis of the host cell, which is being used, uh, we can divide them as vectors for bacteria, vectors for yeast, vectors for animals, vectors for plants. And on the basis of the cellular nature of the vectors of the host cell, you can consider them as prokaryotic vectors as well as eukaryotic vectors. So let's deal with cloning vectors and expression vectors. A cloning vector is a small piece of DNA which is used to introduce a foreign gene of interest into the host cell. And it can be stably maintained within the host organism. But the role of an expression vector is not only just to introduce a gene of interest into the host, but also to express that particular protein encoded by that gene so that it becomes expressed in the new hosts. And a cloning vector is used to obtain multiple copies of a foreign gene of interest. And you can see that if it is in the case of an expression vector, it is mainly used to obtain or analyze the gene product, which may be RNA or protein of the inserted gene of interest. So when you need to find out the expression of a particular gene or a DNA segment, then you use a expression vector. But only if you just want to find out what is the sequence of it, or when you want to produce multiple copies of a foreign gene of interest, you mainly use a cloning vector. Now, the examples of cloning vectors includes plasmids, cosmids, phages, BACs, YACs, MACs, that is the yeast artificial chromosome, bacterial artificial chromosome, and all that. And expression vectors, only plasmids can be used as expression vectors. normally. And a typical cloning vector usually consists of an origin of application, replication, unique restriction sites, reporter genes, and antibiotic resistant site. And an expression vector usually consists of regulatory elements like enhancers, promoters, termination sequences, transcription initiation site, translation initiation sites. And you can see different elements which will regulate the expression of the particular gene of interest in an expression vector. Expression vectors, as we see, they are widely used in the pharmaceutical industry for the production of peptides and proteins like insulin, antibiotics, growth factors, etc. And they are also employed to produce genetically modified organisms and transgenic plants like golden rice and insect resistant plant varieties, etc., which we had mentioned earlier. So I guess you got an understanding what is a cloning vector and an expression vector. When you just want to make multiple copies of the gene of interest, that is called cloning vector. But if you want to get a better expression of that particular protein, then you will use a expression vector. So I will just give you a basic example. Now consider you want to produce a green pigment in your body, okay, at a very high level, okay. Then we use a expression vector. But if you want to just know what is the particular sequence of that green pigment in your body, you use a cloning vector. So that is the difference. That is, if you have to produce high amount, or if you want to find the protein or the RNA which has been associated with that particular gene of interest, then you use a expression vector. I hope you got an understanding or a basic outline of what is a vector, what are the essential features of the vector, and what are the types of the vectors. We will be continuing in the next session. Thank you for now.